Hello friends, my topic is ecological tools and techniques. Ecological studies require the use of several tools and techniques for a proper analysis and a clear understanding of various aspects of population, communities and ecosystem. These techniques are different for plants and animal population. Now we are going to discuss about the techniques that are used in the animal population. The first method is sampling of animal population. Sampling is the selection of representative subset from a whole population. Sampling enables the statistical analysis of population based on the study of samples drawn from them. They give the accurate picture of the respective population. The values that are obtained from the study of entire population are called parameters, and the values that are obtained from the analysis of the sample of population are called statistical values, and the conclusion drawn from these values are called statistical inferences. Sampling methods are of three types: random sampling, non-random sampling and systematic sampling. Random sampling. Random sampling is the method in which the selection of sample units is absolutely a matter of chance or probability. So, random sampling is also called chance selection or probability sampling. This is the commonest sampling method. In it, all members of population have equal chance of getting selected. At the same time, non-random sampling is a selection depends upon the judgment of the selector. In this, all members may not get the equal chance for getting selected. Random sampling is also of two types: stratified random sampling and simple random sampling. In stratified random sampling, the population is divided into strata and the random samples are selected from each stratum and then pooled. Stratified random sampling is particularly useful when there obvious heterogeneity in the population, communities, areas, etc. to be investigated. In simple random sampling, the population may not be divided into strata. Systematic sampling. This is the method in which the population is at first ordered according to some criteria, then the first individual is selected from it and at random, and lastly further individuals are chosen at specific intervals. Comparing to random sampling, it is less reliable. Next method is trapping and collection of animals. Collection of sample animals from their natural habitats is an integral part of ecological and ethological studies. It offers opportunities to study the habits, habitats, morphological and adaptation of animals, to directly observe their behavior, to learn interaction between animals and also between animals and their environment and so on. Prerequisite for animal collection: knowledge about the laws and regulations which prohibit the collection, killing, or domestication of some animal species which may be listed as rare, threatened, endangered, or vulnerable. Knowledge about the animal species which enjoy the legal protection and so their capture is punishable offense. Knowledge about the habits, morphology, mode of life, and the behavior of animals proposed to be collected. Close familiarity with the collection spot or areas where the intended animals live in large abundance. Knowledge about the collecting devices or instruments and expertise in operating and handling them. And knowledge about the appropriate techniques of animal narcotization, killing and preservation, etc. Basic norms for collecting organism. First one is always observe courtesy code. Then always obtain prior permission from the landowner for collection. Consult the expert of the local college, university, natural history or natural conservancy about to the details of the collection. Never remove organisms from their natural habitat or destroy them unnecessarily. Leave the habitat of organism as undisturbed as possible. Keep different specimens separate from each other while taking them to laboratory for identification. Third one is anesthetization, killing, fishing. The killing and preservation of many invertebrates are simple and they can be accomplished directly with the help of strong alcohol or formaldehyde solution. However, killing and preservation of many animals requires some preservation treatment such as anesthetization or narcotization and fixation. Anesthetization. Many invertebrates are highly contractile and they have to be preserved in the maximum relaxed condition. This calls for their slow and gradual anesthetization or narcotization until death or until they are so intensive or unresponsive that they can be killed in extended state with the help of laxatives or preservative fluids. Fixation. Fixation is a chemical process that intends to lock tissue, cells, and subcellular components such as close possible to its original status, even though such is virtually impossible through standard procedures. The most commonly used chemical fixatives are formaldehyde, ethanol, acetic acid, picric acid, mercury chloride, osmium tetroxide, and statement solution, etc. This is all my topic. Thank you.